पिकअप लेके आ गए थे, माल लेकर सब्जी वहाँ रखे हुए हैं। सब्जी उन्होंने देखे जाते हैं कि
तो आ ही जाएगी ना, 10 मिनट तो आ ही जाएगी ना।
वो आती में गए
कहाँ?
हमें कंपोस्ट वाला बड़ा लोग
तुमको बोल दीजिए आ जाएगी, फोन कर दीजिए, मैं फोन कर देता हूँ, वो आ जाएंगे। अभी गाड़ी को रुकने के लिए इसका इतना जाएगा और इतना जाएगा, यही ना तो है ना
मटेरियल तो है।
अरे थोड़ा रुकने चाहिए, एक आध घंटा, कोई जरूरत नहीं। तो हम लोग एजो स्पाइरलम देख रहे थे ना, इज इट विजिबल?
यस सर।
ओके, तो हम लोग ये क्लासिफिकेशन भी शायद देख चुके थे, जिसमें एंजियोस्पर्म belongs to the family Asparaceae, and there are 4 species, that is Angiospermum leproferum, Angiospermum brasiliensis, Angiospermum amazonensis, Angiospermum haloparafrens, or Ceropediceae. But in Indian tradition, Angiospermum brasiliensis are more important, and they are abundant in the fields also. And during the isolations, Angiospermum brasiliensis are mainly found. Okay, so as I have told you, Angiospermum is a mainly a spiral shaped bacteria. These are long in length, and when they are moved, there are a spiral structure will be created. That's why due to these morphological characters, they are called azoospermum. Azo for nitrogen fixation, aspiralens means they are a spiral in morphology. So physiology is nature chemoheterotrophic and associative. Associative means they are associated with the grass roots or paddy roots also. So they are on the rhizoplanes mainly. They are associated at the rhizoplane on the surface of the roots mainly. Carbon source are organic acids, L-arabinose, D-gluconate, D-fructose, D-glucose, sucrose, pectins. All these carbon sources are important for their food materials, and nitrogen source, that is nitrogen through fixation they take, and amino acids, some ammonia, nitrates may be taken up by the angiospermum. Respiration mainly aerobic, and they are comes under the microaerobic groups. Very less oxygens will be required, 0.2 atmospheric pressures uh, uh, oxygens are required. Growth media, nitrogen free bromothymol blue medium will be required, and in this medium there will be 5 to 7 hours of the incubation period is required for doubling. But in ammonia containing medium, where the nitrogen is available for the angiospermum, only 1 hour multiplication time is there. So the doubling time is 1 hour on a ammonia containing medium, whereas 5.5 to 7 hours on a mylate containing semi-solid medium. These are the benefit, about 20 to 40 milligram nitrogen fixed per gram mylate under laboratory condition, that is equivalent to 20 to 40 kg nitrogen per hectare. Okay, so in the C4 plants, mainly rice, sugarcane, maize, millets, jowar, bajra, and several millets also, small millets. They are able to fix 20 to 40 kg nitrogen per hectare per season. Okay, so there is also reports for the increased mineral and water uptake, root development, vegetative growth, and crop yields also. These are recommended for rice, millets, maize, wheat, sorghum, sugarcane, and co-inoculant for the legumes also. Where they are used for the legumes, there is additional nitrogen fixation will be takes place. Response, there will be average increase in yield up to 10 to 15% will be takes place. Now the phosphate solubilizing bacteria, also there are several phosphate solubilizing bacteria, but the Bacillus and Pseudomonas species are predominant, and also fungi are also there. They are more efficient than the bacteria like Aspergillus avamoi, Aspergillus fumigens, Aspergillus flavus, Penicillium digitatum, Penicillium liliaceum. Okay, so these are the fungi mainly. So bacteria, among the bacteria, that is Bacillus megaterium, Bacillus phosphaticum, Bacillus circulans, Bacillus subtilis, Bacillus polymingia, and among Pseudomonas, Pseudomonas striata, Pseudomonas liquefacens, Pseudomonas putida are belongs to Pseudomonadans family, and they are able to fix more, solubilize more and more phosphorus from the soils. Mainly, uh, mainly bacteria are mainly used for the phosphate solubilizing bacteria, bacteria mainly used for the culture or carrier-based materials, and they are able to solubilize a huge amount of phosphorus into the soil. Some streptomyces also solubilize phosphorus significantly, but only bacteria are mainly used. In the case of fungi, they are aerobic in nature, they generate more biomass into the rhizosphere, and uh, there are some disease causing these bacteria, aspergillus and penicillium. There are many these fungi are causing diseases. So there are chances of infection into the plants, and so that's why the fungi are mainly avoided for the culturing of the biofertilizer. Now, PSM means phosphate solubilizing microorganisms in which bacteria and fungi both are comes into this group. So, if you see the morphology, then for the microorganisms, bacteria, it is 1.1 to 2.2 micron size, micrometer, trans, uh, of clearing. Uh, Clearing will be formed, and they are mainly rod shaped, and around microbial colonies, there will be transparent zones of clearing will be found, and that is indicate the extent of phosphorus solubilizing. Uh, there may be a spreading type of growth, and a spore formulation for the bacillus will be takes place also. If you see the gram reactions, then the bacillus are mainly gram positive and pseudomonas are mainly gram negatives. So bacillus polymingia, pseudomonas striata, both are there, and bacillus polymingia and are gram positive, whereas pseudomonas are gram negative. In case of fungi or aspergillus, colloidal heads colored will be colored, colloidiophores are pitted, often appearing rough in general. Air is separated from the substratum. These are the classification on the basis of morphology of the hypha, and there another asexual spores, 400 to 700 or 1000 micrometer long or 5 to 15 micrometer in diameter. For penicillium typically is single particles of fillets that born or consisting of three or more series of the verticals. So these are the mainly morphological features of the fungi, which are you may study in the pathology also, pathological course of the plant pathology also. If you see the physiology, bacteria are mainly aerobic in nature. They are heterotrophic. Their carbon requirement is fulfilled by glucose, that is the main carbon source, but they can utilize other carbon source also. So there is quite variations in the carbon source also. In case of fungi, most molds are strictly aerobic. So most of the fungi are aerobic. Glucose is a suitable carbon source, but other sugars can also be including complex carbon compounds can be utilized by these crops, these fungi. So all these organisms can be grown in a peak of sky media. So media for these are peak of sky. Contribution, they can contribute up to uh, 50 to 20 kg phosphorus per hectare in the solubilizing capacity. So these microorganisms possess the ability to bring the insoluble salt phosphate into soluble form by secreting several organic acids. Okay, under favorable condition, they can solubilize 20 to 30% of insoluble phosphate, and crop yield may increase by 10 to 20%. So uh, there will be solubilization of insoluble phosphorus or the phosphorus which are applied earlier in the fields which remain insoluble into the soil, that are mainly solubilized by these microorganisms. They make it soluble by secreting organic acids in the surroundings, and these organic acids are able to solubilize the inorganic phosphorus and make them available to the plants. ये मोड ऑफ एक्शन है। तो जितने भी बायो फर्टिलाइजर्स है इन द बेसिस ऑफ कैरियर बेस्ड क्लासिफिकेशन, राइजोबियम इज द मोस्ट important, and the mode of action is symbiotic nitrogen fixation. They entered into the root nodules, plant roots, and they form nodules and symbiotic relationship established, and through which they their food requirements are met by the photosynthetic materials or six carbon compounds which are available in the form of glucose or other sugar source, and they are supplied to the bacteroids, and after fixation the bacteria fix atmospheric nitrogen and they supply in the form of ammonia to the plant system. Okay, so host plant are mainly legumes that may be pulses, oil seeds, some fodder crops are also, some spices are also there. Okay, but method of application, that is seed treatment, is the most important method for the inoculations, and they are applied at the rate of 200 gram per 10 kg seed or 1 kg biofertilizer per hectare. So the requirement is 1 kg per hectare, and the seed treatment is 200 gram per 10 kg seeds. The remark is they leaves residual nitrogen, so second crop can be benefited. After harvest of the crop, they 20 to 30% of the nitrogens are available to the second crop. So the soil fertility will be elevated or increased after the taking the legume crops. In the same way, biofertilizers are also there. Azotobacter, same type of dose will be required, and mode of action is non-symbiotic nitrogen fixations. They remain in the rhizosphere and around the roots only. And the host plants are mainly cereals, millets, cotton, vegetables, flowers, and fruit crops also, vegetable crops also. Okay, ornamental plants also. Method of applications is seed treatment mainly. In the same way, about 200 gram per 10 kg seed or 1 kg per hectare. Okay, and remark they also control certain diseases due to the they have some property. They synthesize some growth hormones, some vitamins, some growth factors for the plants, and due to these they support the plant growth and control certain diseases also. Yeah. So this is for Helicobacter. So the mode of applications are similar to the rhizobium. So in all the cases of the carrier-based biofertilizer, the mode of applications are similar. Now the azoospermum, their associative nitrogen fixation will be there. Host plant is non-legumes like maize, barley, oats, sorghum, millet, sugarcane, rice, mainly C4 plants. Method of application is seed treatment. But in case of uh, sugar, sugarcane and rice, there will be uh, seedling treatment will be there, or root treatment will be there, and set treatment will be there. In the case of sugarcane, set will be treated, and in the case of rice, seedling roots are mainly treated. Okay, so method of application is seed treatment, that is 200 gram per 10 kg seeds or 1 kg uh, per hectare. In the remark, it is also able to secrete plant growth promoting substances and can be co-implemented with the legumes also. This is the phosphate solubilizing bacteria. Mode of action is phosphate solubilization. Host plant is soil application for all crops. Method of application, that is seed treatment. Here also uh, the same rate, that is 200 gram per 8 to 10 kg seeds or 1 kg per hectare will be required and can be mixed with the rock phosphates. Uh, After carrier-based biofertilizers, blue-green algae, azula, and mycorrhiza are non-carrier-based biofertilizers are there. Okay, so in case of blue-green algae, mode of action is non-symbiotic nitrogen fixation will be there. Okay, and host plant is rice, mainly in the rice plant because they are required for they require water for their growth. Okay, method of application is soil applications and they are applied at the rate of 10 kg of soil based flax, soil based blue green algae flax mainly used. So, rate of application is 10 kg soil-based flax per hectare are added in the standing crop. When rice crop is transplanted into the fields, and after 10 days, when the rice crop is established into the fields, you have to water the fields. You have to maintain 5 to 10 centimeter water into the fields. At that time, 10 kg soil-based flax of blue-green algae are mainly inoculated or top dressed into the fields with the half kg of the single super phosphate that will increase the growth and multiplication of the algae into the fields and they reduce the soil alkalinity and have growth promoting effect also. So they this there is growth effect. Okay, so these are the remark. Reduce soil alkalinity. So in alkaline soils, they reduce the alkalinity. Also, they absorb sodium from the surface and they cover the surface also with these uh, blue green algae. Has growth promoting effect due to synthesis of plant growth hormones into the surface soil, and that's increase the uh, benefits on the rice crop. Another important uh, biofertilizer for the rice crop is a azula, that is a water fern. So that's also required water for their growth. So mode of action is symbiotic nitrogen fixation, and host plant is rice, and method of application is soil application at the rate 1 ton dried material per hectare, or at least 500 kg dried materials per hectare will be required for complete effect, for the complete benefit, for getting the full benefits from the azula. They reduce soil alkalinity also and has growth promoting effect also. Okay, so in the rice fields, it is also applied in the same way. In the fields, when rice crop is established, the rice seedlings are transplanted. So after 1 week, when the plant is established, then there will be then we will spread the dried azolla into the soil at the rate of 500 to 1000 kg per hectare. It will be multiplied into the fields, and the multiplication rate is very fast. In every 3 days, they will be doubled, and in the 20 days, whole field will be covered by the azolla beds. And then after decomposition, they will contribute a significant amount of nitrogen to the rice crop. So in that way, we can save 40 to 60 kg, 40 to 50 kg of the nitrogen per hectare. That is equivalent to 100 kg urea in the rice crop. Another biofertilizer, that is fungi, that is mycorrhiza. Myco means fungus and raja means root. Okay, so fungi of roots. So in every plants there will be infections of these mycorrhiza. These fungi are there, and due to the mycorrhiza infections, there will be symbiotic association will be established with the plants, and in the uh, and they are able to supply more phosphorus, more nutrients, more iron to the plants. Host plant, many tree species, vegetables, flowers, and ornamental plants, sorghums are more important plants for the beneficial effect of mycorrhiza. Method of application, that is soil application, and method of application is mainly soil applications, or at the time of transplanting it will be applied, and it is applied at the rate of uh, 5 to 10 kg per hectare. At the rate of 5 to 10 kg per hectare. Okay, they are mainly placed below the roots when the crops are transplanted. Okay, or they may be utilized with the compost also. They are mixed with the compost and incubated, and then it will be broadcasted into the fields, or side placement may be done also. Okay, so usually seedlings are inoculated, or at the time of transplanting, these are mainly utilized. Now, these are the seed treatment concept. So, in seed treatment concept, a FIR concept is mainly followed. If for the fungicide, so first treatment will be done if you have to treat the seeds with the different chemicals also. So first you treat with the fungicides, then you have to treat with the insecticide, then you have to treat with the rhizobium culture or bacterial culture. Okay, so in the seed treatment are mainly done with the sticker solution also. So make a solution of 25 to 40 gram jaggery in 400 ml water and mix gently with the seed for half acre area. Okay, so the seeds are then sprayed in shade and used immediately when they are dry. Okay, तो ये सीड ट्रीटमेंट का तरीका है कि जब भी आप सीड ट्रीटमेंट करें तो इफ यू हैव more than one treatment with a chemical, then first you go for the fungicide treatment, then you go for the insecticide treatment. After that, you have to go for the rhizobium treatment. Okay, so make a solution of 35 to 40 gram jaggery in 400 ml water and mix gently with the seeds for half acre. Okay, uh, 200 gram biofertilizer gently mix seed for half acre area. Okay, so that's why for the 1 hectare, 1 kg biofertilizer will be required. The seeds are then sprayed in shade for air dry air dry, and used immediately for the sowing. Okay, so these are the seed treatment practices that will be followed by the all type of biofertilizers, that is carrier-based biofertilizer. In case of rhizobium, azotobacter, azoospermillum, and PSV, these methods are really suitable for the seed treatment. Then for the root treatment also, there are certain methods for the root treatment. For rice crop, a bed is made in the field and filled with water, or you may take a tray, a big tray you have to take that will be filled with water, and you have to add biofertilizer in that and mix them with water, and then dip the roots of the plants in these culture slurry. Okay, and after 8 to 10 hours, or at least 2 hours, you have to keep them there. You have to transplant them. So in that way, the biofertilizers are attached on the roots surfaces, and they will be attached to the root surfaces, and that when transplanting is done, it will go into the rhizosphere. Okay, so liquid biofertilizers are mixed in this water, and roots of this seedling are dipped for 8 to 10 hours and transplanted also. Vegetable seedling can also be treated in the same way. The seedling of chili, cauliflower, cabbage, tomato, even for the brinjal, and many other for the many other vegetables, seedlings will be treated in the same way as in the case of rice seedlings. Now, so for the soil treatment, you have to take around 4 kg of liquid nitrogen biofertilizer, and it is mixed with the 200 kg of the well-decomposed compost, and that is free from the inert materials, and that then it is incubated for overnight, or it may be 2 days, 2 nights, or for the 3 days. Okay, so during this period, the cultures will be mixed with the compost, and the whole cultures are converted into the carrier-based biofertilizer. So the mixture is then incorporated in the soil at the time of sowing or planting, or that may be some amount of the decomposed or placed at the head of the plants, and then the earthing of the soils can be done. Okay, so in that way, these biofertilizers are placed on the root zone, and then irrigation may be done for the proper utilization of effect of these biofertilizers. Then another one is the liquid biofertilizer. So all the bacterial cultures are now available in the liquid form also. Okay, so there is application flexibility with the liquid biofertilizer. Their amount will be required in a small amount. Only for 1 kg 5 to 6 ml of the liquid biofertilizer will be used. So for 10 kg only 50 to 60 ml will be required. So for root treatment, 250 to 300 ml diluted in 70 liter water, and then it is used for the root treatment. For the soil treatment, 250 ml diluted in 10 liter of water and mixed with the compost, and 250 kg compost can be used for the 1 acre. Okay. For the irrigation water treatment also, 250 to 300 ml each inoculum per acre water with various small holes kept in the irrigation channel also. तो आप इस तरह से भी कर सकते हैं, irrigation water treatment भी कर सकते हैं. You have to main emphasis, you have to place the fertilizer and the bacterial culture to the roots of the plants. So how you can spread them in the right way. So you may do through the root seed treatment, with the root treatment, you may apply by irrigation water also. Okay, so where the delivery will be there, or where the water is going to the channel, you put a bottle with a small holes in the bottom, and that will be very small amount of biofertilizer liquid forms that when it comes out into the water, that will be sprayed in whole fields. Okay, so in that way, during the irrigation, you may apply the biofertilizer also. In the case of drip irrigation, you may apply the biofertilizer, liquid biofertilizer into the drip irrigation system. There will be one tank there. In that tank, you apply 1 bottle of the liquid biofertilizer, and that will be through the drip system, drop by drop, transportation of water will take place into the roots of the plant. And through these waters, these biofertilizers comes into the root, comes around the roots of the rhizosphere. These liquid biofertilizers have high demand in Maharashtra and Gujarat, and they have high shelf life also, so it's 1 year shelf life in the area. Okay, so there is great importance of the liquid biofertilizer. ये डोमिनेशन प्रोसेस में नहीं बता रहे हैं, इसको आगे बताएगा. ये स्पेसिफिकेशन आप देख लीजिए. So there are these fertilizers, mainly nitrogen, azotobacter, azoospermline, phosphate soluble biofertilizer, and mycorrhiza are outside of the FCO classification. Okay, so in the FCO they have provided some specifications for their marketing. Okay, so the biofertilizers will fulfill these specifications are only able to sell into the market. Okay, so for the rhizobium culture, base culture carrier based or liquid based, दोनों तरह का हो सकता है. It may be carrier based or they may be supplied with the carrier materials, or there may be liquid based culture. Viable cell count at the time of before expiry, CFU will be 50 million cells per gram. Okay, and 50 million cells per gram of the carrier material, or in the liquids it is 100 million cells per ml of the liquid materials. Okay, so CFA count is one of the important parameters which decided their ability to fix nitrogen or their effect on the crops also. So CFA, minimum CFA means 50 million per gram of the carrier materials of product material, or 100 million cells per ml of the liquid material. So in the liquid biofertilizer, there will be just a double cell count will be there in 1 gram of material. Here 50 million cells per gram. In the liquid, it is 100 million cells per ml of the liquid material. Contamination level, no contamination level for 5 times. 10 to the power of 5 times total contamination नहीं होना चाहिए. pH of these materials must be between 6.5 to 7.5. The particle size in case of carrier based material, all material cells pass through 0.15 to 0.212 mm ISC. So particle size is also very important for their effectiveness. Okay, moisture percent maximum is 32%, 40% in the case of carrier based material. Efficiency character should show effective regulation on all species listed on the packet. Okay, so on all the packets there will be listing of the biofertilizers, what will be the effective regulation, for which crop these nitrogens are there, and the expiry date for the carrier based biofertilizer it is 6 months, and for the liquid biofertilizer at least 12 months. Okay, so the expiry period is Okay, so I have added that expiry period is also there. 6 months for carrier based materials, and at least 12 months for liquid biofertilizer. In the case of electrolyte also, similar parameters are there, like base carrier based or liquid based biofertilizer will be there. Annual cell count, that is CSP minimum 50 million cells per gram of carrier material or 100 million cells per ml of the liquid material. Contamination level, that is no contamination, active for 5 dilutions. pH, that is 6.5 to 7.5. Particle size in the case of carrier based material, that is all materials will be passed through 0.15 to 0.2112 mmISC. Okay, the percentage by weight, maximum in case of carrier based, that is 30 to 40%. Efficiency is the here, in the case of liquid, we have seen that there must be effective regulations in the electrons which are being done from the package. This crop का नाम लिखा हुआ है, और that crop effective regulations must be there. In case of electrolyte, the strain should be capable of exceeding at least 10 milligram of nitrogen per gram of the sucrose. है ना, तो 10 ग्राम sucrose के area में that will be calculated for the fixation of 10 milligram of the nitrogen. Okay, so that is the about 10 milligram of nitrogen per gram of sucrose consumed. That is the efficiency characteristic. Here also the expiry period is there. Same expiry date will be followed. Expiry period, that is 6 months for carrier biofertilizer and at least 12 months for the liquid biofertilizer. Okay. In the same way, US parallel, we may add also. So in case of reverse parallel, the factor characteristics is the same way 50 million cell per gram of carrier material or cell per gram of the liquid material. Contamination level, no contamination at 5 dilutions, pH between 6.5 to 6.5. Particle size in case of carrier material, all materials can pass through 0.15 to 1.2 MIC. Moisture percentage by weight, that is by 30 to 40%. Efficiency characters, that is formations of high leakage in semi-solid plant under free the bromine have been bromine. Okay, so there may be effective formation of white pellicles in semi-solid plant under free the bromine have been bromine can be there. Expiry period is similar to the other carrier-based biofertilizer, that is 6 months for carrier-based and at least 12 months for the liquid biofertilizer. So in the case of phosphate soluble bacteria, all our parameters are remain same. That is base, carrier base, or liquid base. Viable cell count is CF2, that is 550 million cells per gram of carrier materials or 100 million cells per ml of liquid material. Contamination level local contamination from 5 nanomoles, pH 6.5 to 7.5. Particle size in case of carrier based material similar to the other carrier based material, it is 0.15 to 1.2. IC, their moisture percent by maximum in case of carrier based 30 to 40%. Efficiency character, the strain should have phosphate soluble capacity in the range of minimum 30% in the estate of specified in terms of zone formation, minimum high membrane is prescribed area having at least 3 mm thickness will be there. So solubilization diameter will be 5 mm and solubilization zone will be thickness, area thickness will be at least 3 mm. So 3 mm thickness will be अपने 5 mm का solubilization zone होगा. Expiry validity is similar, that is 6 months of carrier base and that is 12 months of the liquid biofertilizer. Total is that is 1 into 10 power 7 CFC per pound per year or 5 into 10 power 7 CFC per ml of the liquid materials. Okay, so these are the totals to be. ये सब तो बात बता चुके हैं आपको. एडवांटेज ऑफ बायोफर्टिलाइजर that is biofertilizers. The nitrogen biofertilizers can provide 45 to 30% of chemical fertilizer equivalent to nitrogen, and TSP biofertilizer can provide 20 kg of P2O5 per hectare per season. Mycorrhiza can provide phosphorus, other micronutrients and help in increase water absorption, which biofertilizers better impact also. Like azoospermide can be integrated with the riboflavin also, and even TSP can be integrated with all the crops also. Which biofertilizers again better control keeps soil biologically active and the biological activity of the soil increase into the processes of the deep bacterial culture in the rhizosphere. They help in soil health. Okay, so these are the advantages of biofertilizer. ये इकोनॉमिक्स दिखाया गया है. तो बायोफर्टिलाइजर का लिक्विड अगर यूज करते हैं तो उसका कितना इम्पैक्ट पड़ता है और क्या कॉम्प्लिकेशंस है. जैसे फॉर द राइजोबियम लिक्विड्स क्वांटिटी ऑफ रिक्वायर्ड per liter 0.1 to 1 liter and cost of application is 40 to 200 rupees and amount of nutrient mobilized that is 30 to 120 kg and that will depend upon the type of legumes. In the case of non-legumes, there is also 20 to 25 hygiene articles there with the cost of 50 to 400 rupees for the purchase of liquid biofertilizer. They are also cost 80 to 400 and they are able to fix 20 kg and 12 to 50 kg of phosphorus. Also similar type of benefits, more nitrogen fixation will be there, and then there will be phosphorus fixation will be there also. In case of mycorrhiza, 2 to 5 kg per hectare will be required by the material. Cost of reduction of 2 to 500 kg and there will be additional 20 to 25 kg phosphorus will be there, and micronutrients and social content will be also improved in the plant system. Less access will be required. ये हम सबको बता चुके हैं कि ये सब प्रॉपर्टी है सभी कैरियर बायोफर्टिलाइजर का सिमिलर रहना है. बेस कैरियर का बायोसेल्स काउंट आपका उतना ही होगा 50 मिलियन सेल्स पर पाउडर और 100 मिलियन सेल्स पर एवरेज लिक्विड कंटामिनेशन लेवल और कंटामिनेशन स्टेट पर 5 लेवल 6.5 to 7.5, partly size pass 0.15, and 0.212 IC, most of the 30 to 40%, और ये सभी safety characteristics है सही है। Riboflavin में should among all the species listed on the packet. Proper नाम packet दिया हुआ है, then must be doses, and riboflavin will be applied only in those parts for they are listed on the packet. Okay, so like testing should be developed 10 milligram per gram of the ages per that information is vital in sensitive that free और phosphate supply by that is at least 30% of the insoluble phosphorus supply, and especially for in terms of Jones phosphorus, there will be 5 insoluble licenses will be created in the staff area of 3 millimeters. तो ये बायोफर्टिलाइजर के बारे में ठीक है, इसमें कोई क्वेरी नहीं होता है।
सर एक चीज पूछनी थी सर, वो इकोनॉमिक्स में बायोफर्टिलाइजर का सर उसमें ये सारे स्टैटिस्टिक्स सर इम्पोर्टेंट है याद करने के लिए, मतलब याद करने के लिए
लेकिन वो याद रखने के लिए पड़ता है, ये सब फ्यूचर में तुम लोग को काम नहीं करते। बोलो जी, अच्छा तुम लोग बताओ कैसे होता है, how phosphate is solubilized by phosphate solubilized bacteria or phosphate solubilized by fungus.
Replacement of hydrogen ions are done by the release of organic acids. Hydrogen ions are replaced. That makes phosphate convert phosphate into available form.
और कोई चलो ठीक है,
ऑर्गेनिक एसिड सब रिलीज करते हैं, फॉस्फेट सॉल्युलाइज्ड बैक्टीरिया मतलब लैक्टिक एसिड, फ्यूमेरिक या एसिटिक एसिड, ये सब रिलीज करते हैं।
हाँ, वीक ऑर्गेनिक एसिड की है,
हाँ वीक
माइक्रोस्फेयर वो अपने माइक्रो रिलीज करते हैं,
यस
है ना, तो जिसके कारण जो भी उसके कांटेक्ट में आएगा उस सराउंडिंग में, उसके माइक्रो एनवायरनमेंट में जो भी इनसॉल्युबल फास्फोरस रहेगा दैट बिकम्स सॉल्युबल, है ना। फॉस्फेटिक फर्टिलाइजर कैसे फॉर्म होता है ना, एसिड ट्रीटमेंट से ही फॉर्म होता है। पहले जो कि क्या करते थे, बोन जो होता था, एनिमल बोन्स को कलेक्ट किया जाता था, उसको क्रश किया जाता था, पाउडर फॉर्म में कन्वर्ट किया जाता था और उसको जो कि है सल्फ्यूरिक एसिड से ट्रीट किया जाता था, तो जो कि है सुपर फॉस्फेरिक ये सुपर फॉस्फेट में कन्वर्ट हो जाता था, है ना। हाइड्रोजन आयंस जो कि है अटैच हो जाता था उससे और उसके बाद वो इम्यूनिटी बढ़ जाती थी। So in the same way, some of the microorganisms have capacity to secrete weak organic acids in the surroundings. That efficient microbes are mainly isolated. They are mainly in the group of Bacillus and Pseudomonas. Bacillus polymelia, Pseudomonas astriata, and Putida, and several fungi are there. They have capacity to secrete these organic acids in the microenvironments, and in that environment, the phosphorus which are present in the insoluble form that will become available, and that's why they are supplied to the plants. Plants are able to take these converted phosphorus in insoluble phosphorus into soluble form. Okay. और एप्लीकेशन बताओ, how root treatments can be done. रूट ट्रीटमेंट कैसे करते हैं
सर? इसमें एक बेड बनाते हैं सर केमिकल का, और उसके बाद 4 से 6
केमिकल
आवर
का
में इसको
नहीं,
रख के देन
केमिकल इसमें तो केमिकल है ही
कौन
नहीं।
सा
केमिकल,
बायोफर्टिलाइजर सर,
हाँ।
बायोफर्टिलाइजर
तो
सर,
तो
मतलब
फील्ड,
इन लिक्विड मीडिया देन 4 to 6 आवर के बाद उसको ट्रांसप्लांट कर देते हैं सर फील्ड में दोबारा से।
हाँ, तो इसको जो कि है ना, या तो आप फील्ड में भी कर सकते हैं, लेकिन अगर जहाँ बहुत पानी की की जरूरत नहीं है जिस क्रॉप में, तो आप एक ड्राम भी ले सकते हैं या एक ट्रे ले सकते हैं। उस ट्रे में आप पानी भर दें और उसमें बायो फर्टिलाइजर ऐड डाल करके उसका स्लरी बना लें, और देन you have to dip the roots of the plants which you have to transplant to the fields. ठीक है। So in the 2 to 4 hours, all the biofertilizers are attached on the root surfaces, and then you may transplant into the fields. That will be helpful for the association of these microbes, है ना। तो उसी तरह से जितने भी ट्यूबर क्रॉप्स है, जैसे पोटैटो हुआ, या फिर एलिफेंट फुट याम हुआ, और बहुत सारे ट्यूबर क्रॉप्स है, बहुत सारे स्पाइसेज हैं, जैसे टर्मेरिक हुआ, जिंजर हुआ, और फिर आपका जो ये है शुगर के क्रॉप्स है, जिसमें बहुत ह्यूज अमाउंट में सीड्स लगते हैं, शुगर के सीड्स लगते हैं। So all in all these cases, you have to increase the dose of biofertilizers, because the planting materials are huge in number, huge in amount. They are used in the quintals, है ना, 5 quintal, 10 quintal, tons. Okay, so you have to increase the biofertilizer quantity as per the requirement of the planting materials. ठीक है। तो जहाँ सीड प्लांटिंग मटेरियल का अमाउंट बढ़ रहा है, तो उसके अनुसार से आप जो कि है सीड का जो अमाउंट है, ये बायोफर्टिलाइजर का अमाउंट है, उसको आप इंक्रीज कर सकते हैं। इसके बाद जो सेकंड चैप्टर है उसको हम लोग देखेंगे, दैट इज बायोलॉजिकल नाइट्रोजन फिक्सेशन इन लेग्यूम्स, है ना। केवल लेग्यूम का ही हम लोग देखेंगे, है ना। सो इन द लेग्यूम सिस्टम देयर इज सिम्बियोसिस रिलेशनशिप विद द राइजोबियम, है ना। सो राइजोबियम इज अ बैक्टीरिया which is found in the soil, but in the soil they are not fixing the atmospheric nitrogen. When they enter into the plant roots, and when the symbiotic relationship with the plants will be established, and the food materials are supplied by the plants as a photosulfate to the bacteroids, then these bacteroids are able to fix atmospheric nitrogen, and they supply to the plants with the good amount of ammonia and amino acids to the plant system for growth and development. Okay, so these are the historical background of the nitrogen fixations in the legume. The classical nitrogen concept of biological nitrogen fixation by legumes has been given by Bossingalt in 1834, and in 1838 atmospheric nitrogen used by the plants, that is legume plants, are established. The facts that the atmospheric nitrogen is used by plants are established in 1838. In 1886, the concept of nitrogen fixation was positively established by German scientists Hellriegel and Wilfart. Okay, so they discovered that symbiotic nitrogen fixation. They given the term the symbiosis process. Okay, in 1888, discovery of Bacillus radicola, now it is rhizobium. It is called rhizobium, that is isolated by the beginning in pure culture. So pure culture of the rhizobium in the form of Bacillus radicola was isolated at the first time by Bidrinki in 1888. Okay, then commercial nitrogen fixation. The commercial history of the biofertilizer starts with the introduction of nitrogen. Nitrogen is a carrier-based materials biofertilizer by Noel Hitler. A laboratory culture of rhizobia in 1895 was done. Okay, so the first commercial biofertilizers comes with the trade name of nitrogen by the Nobel and Hitler as a pure culture of the rhizobia in 1895. In 1951, use of charcoal as a carrier for the rhizobium was given in India. Okay. So the discussion we have also discussed also. So first is the rhizobium is free living in soil and in the rhizosphere of both legumes and non-legumes. So these can be formed in the rhizosphere of they can enter in the symbiosis process only with the legumes, and the nitrogen fixation process starts only when they are entered into the plant roots or legume plant roots only. So in legume root, nodule symbiosis will be takes place. The legume is the bigger partner that is called macrosymbiont, while the rhizobium, that is bacteria, that is very small in size, is the small partner that's called microsymbiont. Two new terms are given, that is one is the macrosymbiont for the host plant and microsymbiont for the microbes, that is the rhizobium. So the rhizobium in the plants is called microsymbiont, while the plants in which the symbiotic process takes place, that is called macrosymbiont. क्लासिफिकेशन आपको बता चुके हैं। राइजोबेसी फैमिली का है, एजो राइजोबियम फॉर स्टेम नोडुलेशंस, जिससे इसमें होता है। ब्रेडी राइजोबियम सोयाबीन, ल्यूपिन, काउपी, मिसलेरी ग्रुप है, काउपी, ग्रीन ग्राम, रेड ग्राम, चिकपी, ग्राउंड ग्राम, ये सभी में है। और राइजोबियम जो की है जो फास्ट ग्रोइंग होता है, जैसे पी, लेंटिल, बीन्स, लेथायरस, बरसीम और लोटस में करते हैं। सो व्हेन ए नोड्यूल बिकम सेनेसेंट, नाइट्रोजन फिक्शन क्या होता है कि नोड्यूल फॉर्म होता है, बट देन द मैच्योरिटी ऑफ प्लांट्स विल बी टेक्स प्लेस, देन the nodule becomes senescent after a period of biological nitrogen fixation. So when flowering stage will be there and grain filling stage will be there. From that period to the maturity period or harvesting period, the senescent period of the nodules will be started. Okay, so decay of tissue sets in liberating motile uh, forms of rhizobium in soil, which normally serve as a source of inoculum for the succeeding crop. Okay, so veil seed formations starts in the pods. The nodules nodule effectiveness will be decreased and senescence of the nodules will be takes place, and when the maturity of the plants will be takes place, these nodules will be decayed. Decay of tissue of these nodules will be takes place, and during the decomposition they liberates the bacteroids into the soil, motile forms of rhizobia into the soils, and these rhizobia are then only act as a normally serve as a source of inoculum for the succeeding crop. Okay, so that's why when you are taking continuously the good crop, so rhizobia will be remain into the fields for the inoculation into the second crops. Okay, ये मार्कोलॉजी मैं आप लोगों को बता चुका हूँ, दोबारा बताने की जरूरत नहीं है। ग्रोथ मीडिया मैंने दो लिखा है, एम और एमा लिखे हैं, और एक और है ये एम और एमा क्या है जी? व्हाट इज एम एंड एमा? ग्रोथ मीडिया
extracted
क्या चीज?
yeast extracted mannitol and
ईस्ट एक्सट्रैक्ट मैनिटोल अगर है ना, तो सॉलिड मीडिया जब है तो वो ईस्ट एक्सट्रैक्ट मैनिटोल अगर है, और जो लिक्विड मीडिया है तो ईस्ट एक्सट्रैक्ट मैनिटोल। ठीक है, ये क्रॉस इनोकुलेशन ग्रुप आप लोगों को पहले भी बता चुके हैं, है ना। ये 1984 में दिया गया था, और ये फ्रेड बाल्डविन एंड मैको का दिया हुआ है। तो इसमें 7 क्रॉस इनोकुलेशन ग्रुप्स हैं, इसमें सबका ग्रुप बना हुआ है क्रॉस इनोकुलेशन ग्रुप, और उसके लिए लेग्यूम्स कौन कौन से हैं। तो जो राइजोबियम स्पीसीज अगर उस राइजोबियम ग्रुप के उस लेग्यूम टाइप के लिए अगर इनोकुलेट करते हैं तो बेटर आपको नाइट्रोजन फिक्सेशन होगा, नोड्यूल फॉर्मेशंस होंगे और बेटर सिम्बियोटिक रिलेशनशिप एस्टैब्लिश्ड होगी। बट वेन you use these rhizobiums with the different crop groups, then there will be different there will be decrease in the nitrogen fixation will be there. Okay, so the principle is the ability of an isolate of rhizobium to form nodule in a limited number of species of legumes related to one another. So all rhizobia that could form nodules on roots of certain legume types have been collectively taken as a species. So it provides a workable basis for the agricultural practice of legume manipulations. Okay, so these principles are mainly associated for the cross enucleation groups, that is ability of an isolate of rhizobium to form nodules in a limited number of species of legume that is related to one another. Another one is the all rhizobia that could form nodules on roots of certain legume types have been collectively taken as a species. तो एक ग्रुप में जितने भी ये legume type होंगे that have been collectively taken as a species. It provides a workable basis for the practice of legume manipulations. That's why farmers can choose a correct type of the rhizobium species for the legumes. Okay. ये राइजोबियम क्लासिफिकेशन ये एल्कान 1984 के बेस पर ऑन द बेसिस ऑफ ग्रोथ एंड डीएनए बेसिस पर दिया गया था, जिसमें जो मेन बेसिस था that is base composition of pure DNA, that is molar percentage of guanine and cytosine के आधार पर दिया गया था. So for fast growing rhizobia, that is the rhizobium, that is the guanine and cytosine content 58.6 to 63.1% and produce acid in yeast extract mannitol agar media, है ना, that is with the fast growing. In the case of slow growing, guanine and cytosine content will be 62.8 to 65.5%. Alkali in the yeast extract will be produced. Okay, so in the slow growing, alkali will be produced. In the fast growing, acid will be produced, and there will be a specific ratio of the guanine and cytosine amino acid content. Okay, so in the fast growing, there will be less content of guanine and cytosine amino acids, whereas in the slow growing, there will be some higher content, 62.8 to 65.5% guanine and cytosine content will be there. ये contributions मैंने बताया था लगता है, फिर भी एक बार देख लीजिए. डायरेक्ट कंट्रीब्यूशन ऑफ नाइट्रोजन symbiotically with legumes, अल्फा में सबसे ज़्यादा है 100 to 200 kg, है ना. फिर पीएलपी में 168 to 200 kg, क्लोवर 100 to 150 kg, चिकपी 85 to 110 kg, काउपी 80 to 85 kg नाइट्रोजन पर हेक्टेयर, ग्राउंडनट में 50 to 60, लेंटिन में 90 to 100, ग्रीन ग्राम ब्राइट ग्राम में 50 to 60, पी में 50 to 77, सोयाबीन में 60 to 80. तो there is quite variations into the nitrogen fixation. Even there is a residual nitrogen benefit for the succeeding crop is there. They are recommended for legumes like pulses, chickpea, pea, lentil, black gram, green gram, cowpea, pigeon pea, or oilseed, soybean, groundnut, fodder crop, berseem, lucerne, spices with methi, and even for the green manure also. Just maybe here. And some have. Okay, there is increase in yield up to 10 to 35% will be there. Okay, now here rhizobium nodule formation will be takes place for the establishment of symbiotic relationship or for the nitrogen fixation. In this figure, I have shown that in the legumes, rhizobia are rhizobia enter into the roots and they forms nodules into the legume plants. If you see here, there is a huge amount of nodules are formed. Okay, so the fixed legume plants supply carbon in the form of malate or sucrose to the rhizobia, and then rhizobia will fix atmospheric nitrogen into ammonia, and that will be supplied to the legume crop for the growth and development. Okay, so there are different type of flavonoids are also secreted into the rhizosphere. I have We have discussed these things into the rhizosphere. Okay, so in case of alpha, luteolin, uh, luteolin flavonoids are mainly secreted in case of clover, geraldone, in case of soybean, genistein. So these crops will secrete these flavonoids in the rhizosphere, and when their concentration is increased more than 10 micromoles concentration into the rhizosphere, then microbes, bacteroids, legume bacteroids are able to identify their host plants, and then the interactions will be takes place for the infections. Okay, so these flavonoids induce interactions at molecular level, gene expression by the signaling to each other. So the concentration of these chemicals, these organic flavonoids are very much important. Their secretion in a specific concentration is important, and when their concentration is more than 10 micromoles concentration into the rhizosphere, the there will be gene molecular level of gene expressions will be initiated, and that work as a signaling that work as a signaling agents or signaling molecules. Okay, so these flavonoids induce interactions among the rhizobia and plant roots, and this will be takes place as molecular level of gene expression by signaling to each other. So these genes which are responsible for the nodulations, are called node genes. These are grouped into four groups. That is, GI is one group. That is possible host range gene, host range genes. They are they identify the root host. CBA is the root hair curling. So root hair curling will be starts or initiated with the CBA genes in the plant roots. Okay. D for regulation. They regulate the process. Or then FELM genes are mainly control of a host specificity. There will be a specificity of a host. There will be cross inoculation groups. So these genes, four group of genes are mainly responsible for the nodulation process, and not gene. This is called not genes, and these genes are indicated by capital letter, that is JI, CBA, FELM. JI is possible host range genes, CBA root hair curling genes, D for the regulation, and FELM for the control of host specificity will be there. Okay. So this is the nodulation process. So this the nodulations will be takes place. So in the first picture, if you see that in normal root hairs are there. Okay. So exudations of organic substances will be there. It takes place. Flavonoids will be takes place, and when the concentration of these flavonoids are increased more than 10 micromoles, then the curling of the roots will be starts, or host genes specifications will be takes place. Molecular level gene interactions will be takes place, and so the normal root hairs. Second one, exudation of the organic substances will be takes place, and accumulation of rhizobia in the rhizosphere will be there through the secretion of the organic acid. Number four, in the fourth one, it is orientation and binding of rhizobia will be takes place. So our rhizobia is binded with the curling of the roots will be there. Acetic acid production will be takes place. Root hair curlings and deformations will be takes place, and formation of infection thread will be established or will be formed. So when the infection threads are formed, the rhizobia are start entering through the Shepherd's crocs. This is called Shepherd's crocs. Okay, so formation of Shepherd's croc cells and entry of infection threads will be takes place, and in the ninth case stage, thread containing bacteroids extending into root hair cells. है ना? So here you can see the thread-like structure will be there, and the rhizobia will be entered into the root hair cells. Okay, so entry of infection thread will be takes place into the cortex, and branching will be done, and nodule development will be takes place. Okay, so in that way, the relations process will be takes place into the plant roots. ये फोटोग्राफ से बताया गया है किस तरह से राइजोबिया का इंटरेक्शंस होता है ना. In the very early events in the rhizobia legume symbiosis, flavonoid proteins inducers flavonoids are mainly secreted and to the that work as signaling molecules. Then the rhizobia, due to the knot factors, they are infection threads are formed into the in the root hair, and the infection will be started. So these are the steps involved, that is flavonoids and isoflavonoids in the legume root exudates is responsible for the host specificity. So several type of flavonoids are there. In case of soybean, genistein is there and isoflavones. So they work as a signal. They signal rhizobia in the soil that a legume is present and ready to nodulates or ready for the infections. So these flavonoids are work as a signaling molecules that signals that the legume is present, a suitable legume is present for you for the infections. Then signaling molecules, that is node factor, released by NIF and node genes and are released by the bacterium upon recognition of the correct host, है ना. क्योंकि जो नॉट जीन्स है उसमें स्पेसिफिसिटी ऑफ द होस्ट भी आपका जो फ्लेम FELM करके जो जीन्स है that is responsible for identification of the correct host. Therefore, the rhizobia, in response to the flavons, release not factors, and not factors are sugars with the specific signaling functions, and these not factors signal to a legume that rhizobia are present in the soil and ready to live with the nodules. So the bacterium get attached to the root hair curlings will be takes place, bacterial invasions of the root hair will be takes place, movement of bacteria to the main root by way of the infection thread will be there, and cortical cell divisions starts and formation of nodules takes place. So after formation of modified bacterial cells, bacteria it will become called bacteroids within the plant cells, and there will be development of the nitrogen fixing stage. So the continued plant and bacterial cell division will be there, and forming the mature root nodules will be there. So for the fixation, NIF genes are also responsible for the nitrogen fixations, and it is called NIF genes. So genes responsible for nitrogen fixations are called NIF genes. In NIF genes, 32 NIF genes are involved and that are arranged in the 7 to 8 clusters. Okay, NIF, QB, AL, FM, Z, WV, SUX, NE, YT, KDH, CJ, है ना. तो इस तरह से NIF जीन्स है, इसमें 22 कैरेक्टर्स है इन्वॉल्व है. That is KDH, that controls KDH, that is in the dark black color, that is control nitrogenase enzyme complex. Okay, so KDH is responsible for the controlling the nitrogenase enzyme. इसका डिटेल में नहीं दिखे हैं, अगले क्लास में इसको डिटेल में आपको बताएंगे. ये यहाँ पे जो नोड्यूल को दिखाया गया है, so exactly they are very pink in color. There is a good nitrogen fixation will be there, and the nodules are mainly filled with the leg hemoglobin. Okay, so in that at that times they are pink in the color. Okay, so pink color is leg hemoglobin, a protein that carries oxygen to the bacteroids, that is liquid. Okay. So this photograph shows the physiology of a legume nodules. So here you are seeing the different shades of the red color. So in the middle one, that is the active zones of the nitrogen fixations. Okay, and this one is the senescent zone where the nitrogen fixation activity was earlier found, and in these zones, that is newly infected regions will be there, and low nitrogen fixing ability will be there. and this white zone, that is a plant cell division zone, that is no nitrogen fixation ability will be there. So this is the nodules that is filled with the leg hemoglobins, and bacteroids are filled with imbibed in these leg hemoglobin proteins. Okay, so when these bacteroids are dipped into the imbibed into the leg hemoglobins, then only limited supply of oxygen will be there that can only can feed to the bacteroids, and they did not harm the nitrogenase enzyme. Okay, so nitrogenase enzyme remain active throughout the period of the nitrogen fixations, throughout the period of the growth period, and that's why the nitrogen fixation ability of the legumes are very high. Okay, so these are the cortex area, these are the what vascular tissue, and this is the endodermis. So from where these leg hemoglobin is synthesized, from where leg hemoglobin comes into the nodules, कहाँ से आता है जी? किसको पता है? लेग हेमोग्लोबिन कहाँ सिंथेसाइज होता है? अच्छा, इसके लिए ग्लूकोज कहाँ सिंथेसाइज होता है, जो बताओ? फूड मटेरियल कहाँ से सिंथेसाइज होके आता है?
सर रूट नोड्यूल्स में होता।
क्या चीज?
रूट नोड्यूल।
आवाज नहीं आ रही है
सर रूट नोड्यूल्स।
रूट नोड्यूल्स में तो नाइट्रोजन फिक्स होता है ना, तो यहाँ पे जो ये लेग हेमोग्लोबिन जो आता है वो कहाँ से आता है, या जो इसके लिए फूड मटेरियल है वो कहाँ से आता है? लेग हेमोग्लोबिन तो ऑक्सीजन सप्लाई को रिस्ट्रिक्ट करने के लिए है, या ये कहो कि एक कंटिन्यूअस ऑक्सीजन सप्लाई के लिए, जो कि रिस्ट्रिक्टेड ऑक्सीजन सप्लाई है, वेरी लो टेंशन पे ऑक्सीजन सप्लाई है, ताकि बैक्टीरियाइड को जो कि है ऑक्सीजन मिलता रहे और नाइट्रोजनेज हमारा अफेक्ट नहीं, क्योंकि जैसे ही नाइट्रोजनेज अफेक्ट होगा तो नाइट्रोजन फिक्सेशन अफेक्ट होगा। ठीक है, तो ये जो जो कार्बन मॉलिक्यूल्स हैं, जो सुक्रोज या ग्लूकोज या और भी बहुत सारे के कार्बन सोर्स हैं, sugar source है, that will be supplied from the as a photosynthesis material from the leaves से जो कि है वो नोड्यूल्स में सप्लाई होते हैं, और जो साइटोसोल से जो कि है लेग हेमोग्लोबिन सप्लाई होता है। Cytosol is the site for the synthesis of the leg hemoglobin that will be supplied to the nodules, and they are filled with the leg hemoglobin. Okay, so in the hemoglobin, that is leg indicate legumes, leg for legumes, and hemoglobin is the same hemoglobin as it is found in the human blood also. Okay, so it is hem proteins having a hem moiety attached to a peptide chain which represents the globin part of the molecules. Its molecular weight is about 16,000 to 17,000, and about approximately they have 0.29 to 0.32% of the iron will be there. In the human hemoglobins, they have molecular weight, very high molecular weight is there, about 6600 molecular weight is there. There is a series of peptide chains linkage there. So 4 peptide chain linkage were there. In the leg hemoglobin, there is no chain linkage is there. Okay, so nitrogen fixation is directly proportional to leg hemoglobin content, how much leg hemoglobin content is there into the root nodules, and that's why for the identification of the root nodules, you see only the pinkish color is the most important nodules that have the more high nitrogen fixation will be there. So the function is as a biological value in regulating the supply of oxygen to the bacteroids at optimum level that is conducive for proper functioning of the nitrogen fixing system, that is not harming the nitrogenase activity. Okay, तो अगले क्लास में बताएंगे। एक बार आप लोग भी लेग्यूम में नाइट्रोजन फिक्सेशन अच्छे से देख लीजिएगा, अगले क्लास में डिस्कस करेंगे और इसको और भी इलोबरेट कर देंगे। ठीक है,
सर हैंडआउट्स दे दीजिएगा प्लीज।
हाँ ठीक है।
Excuse me, sir.
हाँ बोलो
Sir, root nodule, root gall and root knot, इसका difference क्या है?
रूट गॉल और रूट नट्स जिसमें देखो जो बैक्टीरिया के लिए जो नाइट्रोजन फिक्सेशन का होता है उसमें नोड्यूल ही कहते हैं उसको। और नट्स और गॉस जो की है वो इन्फेक्शन का वो अलग अलग है, जिसमें निमेटोड्स वगैरह का होता है और कुछ और भी चीजें होती है। लेकिन जहाँ नाइट्रोजन फिक्सिंग की बात आती है वहाँ केवल नोड्यूल्स ही उसका नाम दिया गया है।
एनी स्ट्रक्चरल डिफरेंस, कुछ स्पेसिफिसिटी?
हाँ, स्ट्रक्चरल डिफरेंस भी होगा, लेकिन मैं अभी नहीं बता पाऊंगा, अगले क्लास में मैं तुम लोगों को बता दूंगा। ठीक है,
ओके सर।
और कोई? ठीक है, फिर अगले क्लास में मिलते हैं। थैंक यू वेरी मच।